Yes, thank you very much for inviting me. It's really very exciting. So you can see my slides, yeah? So it's fine.
Yes, yes, everything works.
Yeah, so I will say some words about our group. So we basically are more statisticians than neuroscientists. So we try to do some analysis on mostly on fMRI and EEG data. So we have around 15 people at our group. So yeah, and today I'm going to talk about my recent work that I just recently finished. So it's about statistical model that we applied to EEG data to find some interesting patterns in effective connectivity of children with ADHD versus healthy children. And this is the outline of my talk. So I will start with some introduction and motivation of this work. Then I will introduce you vector-autoregressive model and the Granger causality approach. Then I will switch to mixed effects framework, then to spectral extension of mixed effects model. And then I will go to data application, so ADHD dataset. And then I will finish with results and discussion. So if you have some questions, you can stop me at any time and just don't hesitate to interrupt me. So
yeah, we love to interrupt.
Yeah, sure. So I will start with introduction, and I think your group is kind of very good familiar with connectivity concept. So it's in general divided into three parts. So it's structural connectivity, functional connectivity, or effective connectivity. And this work was devoted to effective connectivity approach. And as many studies show that not only closely located regions in the brain communicating with each other, but also there are distant ones. And to capture this communication and this causal interactions between the brain, there are some models that people use, and the effective connectivity in general is a concept that tries to know the direction and the magnitude of interactions between brain regions. So there are three, I would say, roughly three directions which people do when they want to know about effective connectivity. So they can be model-based and they can be model-free. So first is dynamic causal modeling. This is kind of a classical approach suggested by Friston. And there are a lot of models which are basically on information theory. And there are models which are based on Granger causality. So today I will tell you about how we use the vector-autoregressive model to capture Granger causality of the brain signals. So the motivation to do this research was that many models of effective connectivity have certain limitations and at the same time different difficulty of implementation. So we wanted to have some model that we can quickly code and quickly get the results. And what we did novel, actually, we apply this mixed effects vector-autoregressive model for different frequency bands of EEG signals. So we will get more insights about how the connectivity works at different frequencies of the brain. And what we wanted to get is that we can use some existing code or existing software to implement the model and to get some results very quickly. So I will start with the vector-autoregressive model. So basically, vector-autoregressive model contains with R components. So we have Y1, Y2 up to R, where these R components are actually the brain regions of interest, for example, in fMRI, or it can be the electrodes of EEG machine. So this vector YT, which contains Y1, Y2 up to YR, is regressed towards its own past values and also other components' past values, plus some random noise. So actually we see that like brain signals, they are actually depend on previous previous state of the signal, and also some previous states of other locations, not only the one that we are interested. And for example, for the component that was one time point ago, we get the connectivity matrix. This phi1 is actually R by R matrix, and each component in this matrix corresponds to like strength of connections between these two. And if this coefficient is non-zero, we say that actually, for example, YR, Granger cause Y1. So Granger causality basically means if we include this component into our model, we can predict the behavior of our brain signal better than not including this component. So I think this is more or less clear what I explained. So for each time lag, we can have connectivity matrices from lag 1 up to lag P. And so each component in this matrix shows the strengths of connection between the channels or regions. And then we go from the basic VAR model into mixed effects frameworks. So what does mixed effects frameworks give us? So basically for each participant I, so for each client, for each child that we gather the EEG, we have still R components, but
what are the components? Can you explain what are the components here? Well, not the R components, sorry, but the dimension of Y. What are the time series? A single time series is a sensor, or what is it?
Yeah, single time series is a sensor. So we have time series with resolution of frequency that we have for this channel. And so this Y, this big Y, it's actually contains of R components, and each component is time series.
But it's an electrode.
Sorry?
It's an electrode, the EEG electrode.
Yes. Or for example, it can be fMRI region. So each YR is the region. So we can summarize somehow regional activity, or I don't know, maybe averaging inside the region. And then we say, okay, this region is Y1, this region is Y2, and so on. So for example, if I have EEG cup with 19 channels, I will have 19 components in this vector.
Yeah, but in the region case, it makes sense that you're looking for interactions between brain parts. But in the electrode sense, you're looking for interactions between electrodes, right?
Yes, for interactions between electrodes. Yes,
Okay.
exactly. Yes. And I was talking about the mixed effects VAR model. So here, what we allow this model to do, so first, it's again this summation of different lagged values for each of this component, but at the same time, we allow these matrices, connectivity matrices, be different in each group. So I will explain it here. So for example, if we have two groups, group of healthy children and group of ADHD children, we want their connectivity matrices have some shared structure, but at the same time, it's different between groups. Yeah, so we create a model in which we say, for example, okay, first 50 participants are from healthy group and last 50 participants are from ADHD group. And then depending on the indicator, on the number of our child, we can get different connectivity structure for them. And moreover, so for each participant, we decompose our connectivity structure into two parts. So first part is group-specific fixed part. So the part that all participants in the group share. And second is random part, which corresponds to each participant random fluctuations, which are like, so they are for each participant defined. So when we have this formulation of the model, as a result of our fitting, we will get the following. We will get group-specific fixed connectivity components and subject-specific random effects. So after that, we know that we can do this. We decided to get the filtered signals. So we decided to apply Butterworth filter to extract this common frequency bands that usually neuroscientists get, like this delta, theta, alpha, and beta frequency components from EEG original signals, and apply our mixed effects model to this extracted components. So we extend the model like to have many fittings for each. So yeah, I will tell about this a little bit later. So for each signal, we have now 5 of them. And for each participant, we have 5 components, and then we fit model many times. So yeah, here I just listed what usually people find as a correlation and the activity of the brain at different oscillations. So usually they find that slow waves as delta and theta are somehow connected to sleep, learning, and memory. So the faster oscillations as alpha are mostly to closed eyes state, and the beta is associated with thinking, active concentration, and gamma is mostly correlated with some higher cognitive functions such as memory, attention. And this is how it looks when we extract this component. So the upper signal is a original signal from one electrode. So from one electrode, it's a signal from 2 seconds of data. And then we apply Butterworth filter to obtain these 5 components, these slow waves up to fast waves. So then what we actually did is that, yeah, I just thought about this. What we did, we have our mixed effects VAR model. So for each frequency band separately, using filtered components, we fit this model for 5 frequency bands. And then for each of these 5, we get connectivity structure and standard deviations of random effects. So for each frequency band. Yeah. So now I go to some advantages that we get when we suggested this model. So first is that this model can vary for number of groups, number of lag values, and account for this random part. So account for random fluctuations in the group. And using this model, we can discriminate this connectivity structure between groups and detect some differences or variances of random effects. So we can easily do statistical inference using some t-tests and likelihood ratio tests. And this can be done with existing software in R or in MATLAB. And using this model for different frequency bands, we can obtain new meanings, like we can get new interpretations how the effective connectivity works at different frequency bands. So now I go to the data that we had. So this data was collected by these authors from children, and it's 19-channel EEG recording at 128 Hertz sampling frequency. So the data was pre-processed and was gathered from 53 control children and 51 ADHD medicated children. And for the whole recording, they performed a visual attention task for about 19 seconds. And they, all the time, they were instructed to count cartoon characters. So for my analysis, I didn't use all 19 channels. I used only 11 of them, like I placed them highlighted on this plot, and I used only 15 seconds of data. This was done because of the curse of dimensionality and because of the number of parameters that we get in the model if we increase the time and if we increase the number of channels. So sometimes it can take a lot of time to fit the model. And now, yes. Yes, sorry, I didn't hear you.
How did you select the brain regions in the 15 seconds?
Yes, this is a good question. Actually, we tried different selections, and we tried to select, maybe it's like a cherry picking a little bit, but we tried to select the channels that are mostly active, that are from previous studies that people use for visual attention. So something like this. It was more like artificial, like how we choose it.
But what about the 15 seconds? 15 seconds at the end?
Arbitrary.
Yeah, this is a good question. So because for 90 seconds, they did the same task. The logic was the following. So we decided, okay, maybe the first seconds were not very good because children could be confused what to do or something like this. And the last seconds can also be like a little bit not representative if the children already tired or something like this. So we just cut the middle 15 seconds.
Thank you.
Okay. Yes. So as the implementation results, so I remind you, so what we wanted to get. So we wanted to get some group structure, so which is fixed for all participants in the group. This is structure that participants share, and also some unique components which each participant brings into the structure. So this random component, random fluctuations of this fixed part. So first, I will quickly tell you about what we get here. So here the plots, they're kind of messy, but they are just for like rough reference what is going on. So here we placed only significant connections and the most active from the highest parameters by magnitude. So we extracted the highest and at the same time significant. And we noticed that the upper is for control group for 5 frequency bands, and the lower is for ADHD group, again for 5 frequency bands. And on this plot, it can be more visible what is going on there. So basically, the differences between two groups will like this. So here, from delta, theta, alpha, beta, and gamma, green arrows represent unique connections that people have in control group, but people in ADHD group don't have these connections. And the red arrows represent the connections that are unique only for ADHD group. So here we can see that, for example, for all frequency bands, like there are components that are only unique for control group, and in most cases, we can see that control group has more significant connections than ADHD group. And at the same time, we can see that at the fast oscillations at beta and gamma, we see different patterns. So we see this P3 strange behavior and C4, for example. So, and by doing this, we concluded that in most cases, control group had more significant connections. And I will go to results and interpretations a little bit after I introduce you the standard deviations of random effects. So the standard deviation of random effects is basically a matrix which contains the random fluctuations of connectivity parameters for each of these connections. And here, for our analysis, we used only lag value equal 1. So we looked at data, which is one time point before, and on this axis, we see these lag values, and on this axis, we see our channels. And if we have some higher component here, this means that this, for example, this P4 channel influence on this F8 channel was kind of variable in the group. And this is for two frequency bands, for delta and theta. And the first column is for control group, second is for ADHD group, and the third is the difference between them. And then this is for fast oscillations. So here is more interesting. So here we can look a little bit closer, for example, at beta and gamma band. So we see that if we look at difference between them, we see that, for example, this P3 channel for beta and gamma band is kind of very variable in control group compared to ADHD group. And the similar thing is for this O1, O2 channels, the occipital region channels. And again, they are more variable in control group. So let me just conclude what we get. So we get that the control group has more effective connectivity connections in all frequency bands, and especially in low frequencies. And these differences in fixed effects are more concentrated in connections between frontal part and parietal-occipital part, which was like found correlated with some cognitive control and attention. So the variability of random effects is also higher for control group, and especially for the connections of occipital and frontal and parietal and frontal channels in slow waves. And also we saw that the different channels influence on this parietal and occipital channels is higher in control group, also at fast frequencies, at beta and gamma. So to conclude all of this, so the finding of this parietal-occipital brain region connection is associated with control and selective focused attention. So it's kind of logic that we get it because children did this visual attention task. And these results are consistent with decreased anterior to posterior connectivity. It was seen in these green arrows, which are more present in control group than in ADHD group. And also results are consistent with finding that ADHD patients sometimes showed the reduction in slow waves across the whole brain compared to healthy controls. And so I think that's more or less what I wanted to show you. So if you have some other questions, yeah, I'm glad to answer.
What type of correction did you use for the multiple corrections of your p-values?
So for the p-values, we did I think we did just Bonferroni correction.
Across like all lags or just within one lag?
These results are only for one lag, for lag equal to 1, because we did actually we did the exploration. So why do we use only one lag? Actually, this was done not only because it's faster and we can quickly get the results, but also because when we did analysis for like best lag value, which is like best lag selection according to some information criteria, we found out that the best lag value was 4. So actually, ideally, we should include 4 lag values into our model. But if we look at the matrices of coefficients, if we include all 4 lags, we will see that actually most non-zero parameters are on the diagonal. So actually, this means that the channel's previous information influences on its next information through the different lags. But we are not interested on its own influence. We're interested in cross-regional influence. And because when we go to up to 4th lag and we don't see much non-zero connections for 3rd and 4th lag, and even for 2nd lag, most of them are on diagonal. So we decided, yeah.
Couldn't you constrain the matrix to be zeros for like the 2nd, 3rd, and 4th lag? Because my personal hypothesis would be that there's different lags between different regions, right?
Oh, okay. So so when we fit the model, we use this. We don't fit the model for each region separately. We fit the model for all regions of selection. Yeah. So they are. Okay. Yes. So I meant that, for example, for mixed effects model, we did this per region. So we choose a region and then create a matrix for it, create a model for it, and then fit the model per each region. But when we did the model selection, so we actually fit the model for all of the regions that we selected. That's why it was like finding the best lag value for all regions.
So I guess my question is, like, if you would constrain the lag larger than 1, if you would constrain the diagonal to be 0, right, you would only get cross-region terms. I think if you fit your model with multiple lags in that case, where you constrain it, you would maybe find that different regions have different lag values for which they have larger off-diagonal values.
Yes, probably. Yes.
Okay, that was my question, because I've noticed that in my own work a little bit, where like different regions have different, even at different times of different.
I can address your problem, how it can be done differently, how it can be done within the system differently. If you assume that your system is fully observed.
Yeah, that's a more process. Yeah, then you said lag 2 all the zeros or something.
Well, then you can have a higher lag, you can have longer information transfer time between different regions, but you have to transfer through somewhere. So there will be longer path. That's it.
No, but I don't with EEG for sure. Also, you would have multiple, like depending on how large the white matter tracks is, or like how the effect is. It could also just be not even through another region, but like it could just take longer to affect the other region, right? Because EEG's temporal resolution is super high.
Yes, I agree. Yes.
So it doesn't even have to be through other regions. That's for fMRI, I think. But for this case, it's going to be like even just the how long it takes to transport is different for different regions. Because it doesn't even have to be over white matter tract. Some of these regions can just communicate over the cortex, right? And that would be much faster than over the white matter, or like that would be different than over the white matter tract. Not be slower, actually, not faster, but
yes.
so it's very interesting. Yeah, there's like a lot to do here, I think.
Yeah, you know, like this model is very simplified, I would say. Yes. So we tried like the simplest model that we can do. Because when we do this mixed effects, when we do this for all 5 frequency bands, it's already very overloaded. And I think for one channel, we fit it around like 20 minutes for one channel. So it takes time. And more channels we include, like more we wait for the results.
Right. No. I think it's already good. I'm just saying like it is cool because you can really, I think there's a line of work essentially. There's a lot of cool stuff.
Yeah,
Because
certainly you haven't started looking at nonlinear effects because that would just increase your complexity even further. But so you have this, starting with the linear model. Can you use this to then pull those effects out and begin looking at nonlinear effects with a subsequent model?
And okay. So I think maybe this one.
Right, but your autoregressive model it's fully linear, right?
Yeah, it's Gaussian
Yes,
linear model.
it's linear. It's linear. Yes, we
But you
don't
can
have
subtract.
non-linear things here.
That's what I mean. Yeah.
Yes,
You
this
can
is actually a limitation. Yeah, this is a limitation of the model. So we are not able to capture non-linear interactions.
Yeah. So if you were going to do that next, could you take this current model, subtract the linear dynamics out, and then just have, so you wouldn't have to sort of simultaneously learn the linear and nonlinear, you could just learn the nonlinear?
Yeah, look at the residuals
Yeah, exactly.
from this model.
Yeah.
Whatever it
Okay,
may be.
yes, actually, yes, we look at residuals. Yes, we're interested in it because, if you apply a linear model and your residuals are still non-linear, like don't behave like a white noise, like, yeah. So, but for this model, we still know that for some of them, we still can have some dependence in the residuals, and we somehow close our eyes on it. But of course, it's not correct approach. Of course, we should use as much parameters in the model until we get this white noise as residuals.
Interesting. And there wasn't any windowing done here either. So you assume the 15 band is stationary and
You're assuming
yeah,
structure is stationary.
Right. Yeah.
Okay, so I have a question. You started with Granger causality,
Mhm.
and well,
I was gonna make that a polemical discussion as well. 'Cause I think
Yeah,
Granger
yeah.
causality.
and even Granger causality is problematic, although surprisingly well works surprisingly well. But then you jump to the Gaussian linear model, which is. Do you think it's related to Granger causality in a sense? Because you're not doing here.
Sorry, I think you disappeared.
Variable y1 at lag t-2 predicts variable y.
Okay.
Yeah, you did not, or did you? Did you actually apply any ideas from the Granger causality testing here?
So, like, as we think here, like what we mentioned here, that if we get this matrix, like from our mixed effects model, we get this matrix for lag equal to 1, for example. This coefficients, we treat them as Granger causality. But the question here is still like, are these coefficients are significantly non-zero, or they are like, you understand what I mean? Are they just very, very small, or they are non-significant?
I'm not too sure now, but I don't think this is correct. You need to condition on other, you need to filter out, you have a linear model, So you need to regress out all, like everything but the two signals of interest. And when you fit VAR model, this doesn't happen. They're all kind of intermixed together. That's
They're
why
all mixed.
I wouldn't
Yes.
equate this. I wouldn't equate this with Granger causality.
Okay. So yeah, maybe I need to look at this more. But this is what we've done, like based on previous work. So they call it like Granger causality, and we somehow agreed.
Just
Yeah.
kind of like a predictive relationship, right? That's all it is. It's like what they're looking at. They're not doing any.
But Granger causality is actually more involved. It does look at conditional independence. It's something intuition-wise. It's something versus at correlation matrix versus inverse of correlation matrix at partial correlations. So
Okay.
you're not filtering out other relationship at least this is my strong conviction at this point. I need to look
Okay.
further.
Okay.
Any other philosophical or detailed questions?
I mean, I think maybe they're not looking at full Granger causality currently, but they're certainly doing sort of the predictive part that Granger causality does. Don't think pictures. Right, and so that in that sense, it makes sense why people call this, Granger causality, but
Yeah. Well, formally I wouldn't, but yeah, I see what's
Something like that.
no, things like that exist.
And you should, if you're doing Granger causality too, to say something is Granger cause, don't you have to drop out? So if you have multiple variables, don't you have to drop out
You have
variables?
to regress them out.
Yeah, exactly. So that you do have to do, for Granger causality. Did you do that at all?
Not within VAR model, right? I mean, it just doesn't allow for that. You have to do something extra on top of that. Maybe as next step, maybe we'll overwhelm you with suggestions.
Yeah. This is all just making. Like,
Yeah, we're just thinking aloud. That's what we do in this hour.
It's very cool work though.
Why sensors? Like the relation between HD sensors. I don't know. Maybe.
Why didn't you found sources?
Well, with AG, yeah, that's a mess. But something
Yeah, that's
like.
a mess. That's truly a mess. Yeah.
But the relation between electrodes, they're so much more correlated between each other due to the volume currents, due to the skin conductance, and like non-brain related stuff, basically, not that. And that's why I'm kind of.
Yes, of course. Yes, with EEG, this is always the problem. Like, it's hard to understand, like, what is the source of the signal, what is the nature of the signal that we recorded, actually.
Well, they just use MEG data. I don't know.
Yeah. Makes
Yeah,
sense.
well, I'm not sure with MRI. Everything has problems, of course.
Just use ECOG. Just get it.
No, just open up someone's brain and use optogenetic.
Yeah.
Source.
Yeah, at least then you're looking at
You're
something
definitely
relevant.
looking at
Right?
Work with C elegance, yeah.
Exactly.
Right.
And write a paper that's called Do C elegans have ADHD?
Yeah, what are your next steps with this work? I'm curious.
Yeah, we can suggest you everything.
Yeah, but if you already have a trajectory for it.
Yeah, what do you think? Yeah, good question.
So I think as for this project, it's more or less finished because we didn't want to, I would say, make this more difficult and make this fit forever. Like, actually, it would be great if I could do some penalized regression or something that could like increase the computation time, but we decided not to go into this direction. So I think I'm gonna switch to different project maybe, but if you have really something interesting that can be done based on this, I'm really happy to hear.
Are you going to be working more with Granger causality or like autoregressive models? Do you know?
I think now I'm more focused on the nonlinear relationships between the, yeah, so we did some copula-based models. Yeah, so, and also maybe some machine learning, but did not decide yet.
Yeah, you may want to look at Hilbert-Schmidt information criteria and like
Yes.
other kernel independence
Yes.
methods.
Talking about Schmidt. You can also look at Schmidt.
Oh yeah, Schmidt-Huber must be there at your university,
Yes,
right?
Schmidt-Huber is at our university.
Yeah, so you can just ask him for ideas if you want to do machine learning.
I mean, he's probably already solved that for Rise or like ADHD.
No, don't try to ridicule him for that.
Hey, no, I'm not sure.
What he solved, he
But
solved.
it is funny because every time someone has a new idea in deep learning, there's a paper from Schmidt-Huber in 1980 that's like, oh, I can do the same thing.
Yeah, that's funny.
He has a time machine.
Yeah,
That's what he solved. I think that's what he solved. But yeah, he has a lot of great ideas or had back then.
Well,
And published them.
Come on, that is actual ridiculing what you're doing. He had good ideas.
Well, but he published them back then, right?
He still does, I think.
Yeah, lots of stuff like upside down RL is really great. Yeah.
By the way, what is the time step in this model?
It's 128. Right?
Yes. Like the sampling frequency was 128.
Good question. When you do filtering,
Mm-hmm. Yes.
you're changing the sampling rate because it changes your Nyquist limit. So the sampling rates are different here for each of the frequencies.
Mm-hmm. So I never thought about it. I just filtered them and used them. Like, is it really a problem?
Well, if you
I would
use
call it a problem.
Yeah, you use MATLAB or something, like usually the tools that you use take care of that, the sampling.
Yeah.
In order to
Like-
avoid aliasing, the samples are thrown away.
Yeah.
If you, like for delta, for example, not for delta, for theta, then and alpha, definitely you have fewer time points than for higher frequency. Otherwise-
Yes, sure. Yes, of course. That's actually like this. Yeah, we have fewer time points.
So
Yeah.
there will be slightly, I don't think it's a problem. We're just messing with you. We're just thinking aloud. But it's interesting that you will have different statistical power even for different frequency.
Yes, it's true.
bands.
Will be different.
Mhm.
Yeah.
Okay, well,
Thank you very much.
yeah, thank you very much. That's great. And thank you for agreeing to give this talk on this such short notice. Now you know that we're here and the timing is right. Maybe you want to join every Friday just to hang out with the group. Yeah, we have this reading club every Friday at Eastern US Eastern time at noon. So
Thank you very much for
So
inviting
we
me.
start some paper.
yeah, we discuss papers, of course. Yeah, but somebody presents the paper. Next week, Pavel Popov will present quantum machine learning, a paper in Nature.
Okay, great.
So yeah, you may want to. Let me turn on our camera and so Ksenia can
Yeah. Bye.
say bye.
Thank you very much.
Bye,
Yeah.
thank you.
Thank you. I'm stopping the recording and everything. Oh, I don't know. Okay.